The next thing that we'll talk about is the ventricular system of the brain. Now, the first thing to consider is what the ventricular system is. The ventricular system is basically a collection. It's a collection of fluid within the brain that is formed basically because of the remained patency of the lumen of the developing neural tube. So the brain of it originally develops from the neural tube, and cells of the wall of the neural tube proliferate to form the cerebral hemispheres and other structures. And the cavity of the neural tube doesn't dilate and remains as a ventricular system. So that's basically how the ventricles evolve. In terms of the next thing that we have to cover is the basic arrangement of the ventricular system. What we know is basically there are two lateral ventricles, one in each cerebral hemisphere. These are connected to one central third ventricle by foramina of Monroe, which is labelled here. The third ventricle in turn leads by a cerebral aqueduct, labelled here, to a fourth ventricle which lies just posterior to the brainstem, in between the brainstem and the cerebellum. The first thing that we'll look at is the lateral ventricles, and then we'll look at the third ventricle, and we'll leave the fourth ventricle to consider with the brainstem. So, talking about the lateral ventricles first, the first thing we need to consider is their structure. The basic parts of the lateral ventricle are the anterior horn or frontal horn within the frontal lobe. Then we've got the body, which lies immediately above the thalamus. We have the posterior or occipital horn, which projects into the occipital lobe. And finally we have the inferior or temporal horn, which projects into the temporal lobe. The final thing that we need to the next thing that we need to consider with the lateral ventricles is its position, and we'll consider the position of each of the parts of the lateral ventricle separately. So we'll start with the anterior horn. What we note about the anterior horn is that it has a medial wall which is formed by the septum pellucidum. It has a lateral wall which is formed by the head of the caudate nucleus, and it has a roof which is formed by the corpus callosum. If we move backwards now to the body of the lateral ventricle, we'll note that it has a floor formed by the thalamus, the lateral surface of the thalamus, so the lateral part of the upper surface of the thalamus. It has the floor also has in the medial part the bodies of the fornices. You also have a medial wall, which is once again formed by the septum pellucidum. You have a roof formed by the corpus callosum, and finally you have a lateral wall which is formed by the body of the caudate nucleus. If we now turn our attention to the posterior horn, we'll note that once again there is a roof and a lateral wall. Both of these are formed by fibers called the tapetum, and what the tapetum is basically it's the expansion of fiber. It's the white fibers which pass from the corpus callosum to the temporal lobe. So the corpus callosum lies here, and understandably to pass to the temporal lobe the tapetum has to pass laterally and downwards, and thus they form the roof and lateral wall of the posterior horn of the lateral ventricles. The posterior horn also has a floor, and the floor basically has a calcareous. The floor is basically the white matter of the occipital lobe, but the major feature of note is that the calcareous sulcus extends quite far into the substance of the brain, and thus raises a bump on the floor of the occipital horn, and this bump is called the calcar avus. The next thing to consider is the position of the inferior horn, and the things that we need to note here is that it has a floor and it has a roof/lateral wall. The roof/lateral wall is formed once again by the tapetum, which is labelled here. The roof has the tail of the caudate nucleus associated to it, And the floor, importantly, has a large nucleus related to it called the hippocampus. And this is quite a characteristic feature of the floor of the inferior horn of the lateral ventricle. The next thing that we need to consider is the position of the third ventricle. Now, the third ventricle lies in the midline, and a number of structures line its floor, and we talked about it when we were having a look at the undersurface of the cerebral hemispheres, and we'll revise it once again here. So from front to back, the structures which form the floor of the lateral ventricle, so the floor of the third ventricle, which incidentally is this space here, so that space is the third ventricle here. From front to back, the floor is formed by the optic chiasm, the infundibulum or pituitary stalk, the tuber cinereum behind this, the mammillary bodies, and behind the mammillary bodies we have the posterior perforated substance, and finally the upper surface of the midbrain, before the third ventricle leads backwards to the cerebral aqueduct behind. If we can identify these structures on the undersurface of the brain, as we talked about before, and this is really important because you can actually access the third ventricle through cutting through these structures here. So once again, from front to back, we've got the optic chiasm, the infundibulum or pituitary stalk. We're just going to label the pituitary here. Then the tuber cinereum, the mammillary bodies, and the posterior perforated substance before we reach the midbrain. The final thing that we need to note in terms of position of the third ventricle is its position related to the thalamus. The final thing that we need to learn in terms of the position of the third ventricle is its roof and lateral wall. The roof is formed by a thin sheet of neural tissue called the tela choroidea, and the bodies of the fornices are related immediately above this. The lateral wall is formed above by the thalamus and below by the hypothalamus. And it's important to note the relationship. So you may ask, what's the point in remembering all of these relationships to the third and lateral ventricles? And the point is, if you remember that, you can use the lateral and third ventricles as a guide to identifying structures on brain CTs, which is where your real money's at.